Read the end of the chapter. Who did that? Oh, many hands. So exciting. So since you read, everybody knows what those are. What are those? What code for amino acids? Codons, anticodons. What are they? Each codon can be found where? Correct. Messenger RNA, codon. And a codon consists of what? 3 letters. They're not letters, though. They're not letters. 3 nucleotides. 3 very specific nucleotides with those specific nitrogenous bases, right? Because what are the building blocks of nucleic acids called? What are the building blocks of nucleic acids called? Yo! That's all right. You don't have to know. I'm just yelling at everybody else. That was loud. Nobody? You said you read. Oh, doesn't mean. Okay, this one's from chapter 2. Go ahead. Don't raise your hand. Just scream it out. No. Nope. Bing. Thank you. Phosphate, sugar, nitrogenous base. What do we call those? Little units. Nucleotides. Very good. Nucleic acids are made up of nucleotides. Say it loud for the rest of them. Don't be so quiet. It's comforting to me when somebody will say the right answer, yes? And each nucleotide can be one of 4 if I'm talking about DNA. What are the 4 different nucleotides? Guanine. And what are they? Now I'm going to answer your question. And what is each nucleotide made of? Phosphate, sugar, nitrogenous base. Yes? You with me? Okay. Now I'm glad I know it. Now guess what? Now you have to know it. I already passed A&P. I'm good. Yes? It's your turn now. Okay. So how are you going to... That's a lot of information, right? How are you going to remember this? Just because she said it in class and then I said okay, and okay, I'm good. No. So how would you then put that information down for yourself so you might be able to remember it? You like the flashcard. She's the flashcard lady. I hate flashcards. I hate flashcards. You know why I don't like flashcards? For me anyway. Some people love them, swear by them. My daughter is the queen of flashcards. I have a flashcard company somewhere because I buy her so many flashcards. They work for her. What works for you, keep doing it. If flashcards don't work for you, you know what I do? A couple pieces of paper. Now pretend you're writing cheat notes. Of course, you would never use them in my class. But if I said to you, I'm going to give you 2 pieces of paper to bring with you to class to take your test, what would you do? Yeah, write down, and for that whole nucleic acid thing we just talked about, you'd say nucleic acids made of nucleotides, phosphate, sugar, nitrogenous base. Yes? Just a couple of words. So that's what you do for that. And guess what? If you go to study and you have like 2 or 3 sheets of paper that you go over and over and over again, you're good. You have to make the information small. Not to scare you or anything, but you should already be at that point. Right? Because when's your exam? Tuesday. Okay? So if you're not at that point right now, get to that point today or tomorrow. Then all you have to do is keep going over it. Flashcard, keep going through the pile, right? Because the more you do that, the more it's going to stick upstairs, right? Say yes. Good. So these are a list of, according to the textbook, 3 bases in an mRNA codon. So for every 3 nitrogenous bases in DNA, there's a message. And who's going to take down that message? Messenger RNA, exactly. So if the DNA information says AAA, what would the codon be for that DNA information? It would be U, U, U. Now go up on the chart and find U, U, U. You see it? That's why I picked that one, because it's easy to find. And U, U, U is eventually going to mean what? That's phenylalanine. It's one of the amino acid, PHE. I'm not going to make you memorize that. God, shoot me in the head right now. I wouldn't do that to you. Remember, how many different amino acids can I have? 20 different amino acids. And for that particular one, how many different codes can I have to tell me that that's where that particular amino acid has to go in my protein? There's 2 different ones. You see that? 2 different codons. 2 different transcribed messages that I got from who? From DNA. mRNA, that's these up here. And sometimes you'll see different charts. This one happens to be of codons. Sometimes you'll see charts that say anticodon. Now that is going to be a message or joining of messenger RNA with who? Yes, it is transfer RNA. Now transfer RNA, another member of the RNA family, has what we call an anticodon. And which anticodon will fit to UUU codon on the messenger RNA? AAA. Yes? That sounds familiar, doesn't it? That was the original what? It was the original DNA code. So that is going to join up with messenger RNA, carrying along with it who? The amino acid phenylalanine. So let's just talk about how this works. Transcription, translation. This diagram, your friend. Because on this diagram, we have all the steps. The sequence of events in translation. We're going to start understanding what goes on. So last class I said it's sort of like the big boss sitting in the nucleus, yes? What's the big boss in this case? DNA. DNA has, or is made of, specific nucleotides in a specific order. Is the light starting to go on yet? Is my DNA the same as your DNA? Actually, most of it is, because we're both what? We're both humans. But we have a percentage of our DNA that's different. That's why we're different. Different how? Well, actually, we're very similar. Let me pick somebody else more different. You're more different. Like, you have what color hair? You have blonde hair. I have gray hair, brown hair. Used to be brown hair, kind of brown hair. You have what color eyes? Blue. I have brown. Yes? That's what makes us different. The different portions of our code. You have kidneys, you have a heart, you have a digestive system, so do I. That's the same part of our genetic information. You see where I'm going with this? Good. So that information is a code. That sequence of amino acids is dictated by that sequence of nucleotides in your genetic material, DNA. In order to get the message out to where I'm going to do the business end of making proteins, and that's basically what DNA is, the map for all the proteins in your body. In order to get that information out, I have to make a copy of the genetic material. With the help of a complementary type molecule, something that can fit in and copy the information from DNA, and what's that something I happen to be talking about? RNA. The enzyme is going to help me put that message together. But RNA is going to make a copy of that information, the sequence of nitrogenous bases in DNA. With the help of enzymes, like what? RNA polymerase. So when you hear the word polymerase, think, help me put together nucleic acid. Because we learned that word when we had to copy the DNA to make new cells, right? What kind of DNA, what kind of polymerase was that? It was DNA polymerase. Well, I'm making new RNA, I need RNA polymerase. That's going to help me unzip and pick out the message that I need. Now remember, this is a map for your whole entire body, but I don't want to make every single protein right now. I'm just concentrating on one. So what do? I'm going to unzip information just for that one, at the start point for just that one. Yes? And with the help of RNA polymerase, I'm going to make a copy of that information. Now, in order for me to make a protein, pick a protein, any protein. No proteins in mind? How about hemoglobin? What kind of protein is hemoglobin? It carries oxygen and carbon dioxide in the red blood cells. Question again, what kind of protein is hemoglobin? That might be a question on your test. Nobody knows the answer? I have coffee, I can wait. There's 65 people in this class and nobody knows what kind of protein hemoglobin is. Come on, somebody does. How many? What is it? Somebody said it, I heard it. Say it out loud. Nobody's going to bite you hard. Might be a little bite. Very good. What kind of protein is it? There's four, well, we described different shapes. Is that helping? It's globular. How many globs does it have? This coffee's good. Pumpkin spice. No, nobody knows. Nobody knows. It's a large. I have a lot. It's a quaternary protein. Yes? A very specific shape. Yes? How did it get its specific shape? How do proteins get their shape? Folding themselves all up depending on what? The order of amino acids. Very good. And their size. Because the bigger they are, the more folds we're going to have. It's on page. You don't remember? Oh my gosh, it was such a memorable discussion for me. At least, anyways. Wait a minute. Glucose, lipids. Yeah, there's a beautiful picture on page 49. Yeah. Okay. So the order of amino acids is going to determine the shape and function of a protein. The shape of a protein is going to determine its function, and where does it get that from? I'm taking a long time for this because I need you to understand the connection. You need to understand the connection between DNA, sequence, and protein function. That's why she's got blonde hair and I have brown hair. Do we have the same hair color proteins? No. Why? Yeah, the sequence of my amino acids is different because my DNA code is different. Hopefully the light bulb. Is there light bulbs anywhere going on? Yes? No? Maybe? Sort of, kind of? Give me like a nod, a little head shake, or yes? Good. A head shake. I'm so excited. So, messenger RNA copies the information. And now we have to bring it to the place where I'm going to actually assemble my proteins. Now, proteins are made of what? Amino acids, not nucleotides. Who's made of nucleotides? DNA. Proteins are made of amino acids. Now, how do I get them together? Well, remember, and again, we're revisiting that chemistry chapter, chapter 2. We said in order for chemicals to react, or molecules to react with each other, what do they have to be? Close enough together to do that. And that's what we have to do with the message now. Messenger RNA, a copy of the information from DNA, is then, once it is made, and with the help of enzymes, cut off, it's going to travel outside the nucleus, and we see that in our diagram, newly made and edited, because there's actually enzymes that are going to go in and cut out extra bits and edit. Messenger RNA is going to leave the nucleus and travel to a ribosome. Now, if we look at the cell's makeup, and all of you have learned all the cell parts already, you see that there's a bunch of endoplasmic reticulum that look like they're attached to the nucleus. Doesn't it look like that in the picture? And on that endoplasmic reticulum, there's a whole bunch of little dots. Well, what are the dots? Ribosomes. So we're going to travel to one of those places, and we're going to use the ribosome, which is a 2-unit molecule, to help me knit together my proteins. They're actually going to follow down it like a knitting machine, yes. So, when we get to the ribosome, we're going to see a little docking station. And you can see it here in the diagram. There's an E site, there's a P site, and there's an A site. You see that? And you notice it's going to, the messenger RNA is sort of going to click into that ribosome. It's going to sort of hold it steady so I can do the next part of work that I need to do. The next part of work I need to do is going to involve transfer RNA. Now I have to transcribe the message. Translate the message. Making messenger RNA is called what? Transcription. Bringing together the amino acids to create a protein is called translation. So I'm going to lock in my messenger RNA into my little ribosome. And I think of a ribosome as a little knitting machine. Anybody knit? Nobody knits anymore, just a little bit. You know, you get a couple of needles, got some thread or yarn, call it yarn, right? And the needles are going to help you tie it together, and your product kind of falls out the other side. Well, think of your ribosomes as little knitting machines for proteins. Yes, sir?
The mRNA is it made in a full strand, or is it only made in pieces that's needed?
I don't want to blow your mind or anything, but it's actually made in pieces and connected. So think of it as a strand. One strand of messenger RNA equals one protein. You're going to have a couple of different players knitting things together, and that's the editing part they mentioned. When you take genetics, it's a lot more complex than what we're talking about. There's a whole bunch of other steps involved. But what we want to understand is the basics. Okay? Even this part, making the protein, happens in sections, and then it's all joined together. So, messenger RNA hooks into the ribosome, and we're going to start the process. The process is going to bring the other RNA we mentioned before, transfer RNA, into this ribosome, and it's sort of going to act like an enzyme to help bring things close together so they can do what? React. And the things I'm talking about are what? We're making a protein. Who? Well, that's going to help bring them together. But what do I want to bring together to make a protein? The amino acids. So we see there's a whole bunch of these little transfer RNA units floating around. And with the help of enzymes, enzymes are going to help stick very specific amino acids on these different shaped transfer RNAs. Guess what? Specific amino acids only can hook up with specific transfer RNA based on their what? Based on their shape. The bottom part consists of 3 nucleotides. What do we call the bottom part of a transfer RNA? That's the anticodon. Yes? That's only going to match up with what? The correct codon in the slot on the ribosome. So what I have is a place where new amino acids can come in and hook up with ones that are already there, transfer RNA. The P site and the A site. What's the E site? Yeah, it's kind of like the exit. Push it off. What's going to stay behind is what? The amino acids. Because see those little balls at the top? So see that little guy over there? We see the anticodon there waiting for a transfer RNA to come in. A, U, C. What would be the anticodon that would fit into that spot? U, A, G. I don't know why they didn't make that a G. It would have been nice. Say that was a G over there. Pretend it was a G. Oh, okay. UAC's in the, yeah, I got you. You're right. So the next one's going to come along and do the same thing. Hook up. It's going to have a different amino acid. You got it. And when they come in close enough together, what's going to happen to the 2 amino acids? They're going to bind. And then we're going to break away from the transfer RNA. The transfer RNA is going to, the whole thing is going to move over one, and the transfer RNA is going to get booted out, leaving behind its what? Amino acid. And it's going to keep doing that until we get through the whole what? The whole strand of messenger RNA. So what we leave behind is kind of like a chain of pearls, which is actually a chain of what? amino acids in an order based on the genetic code. The messenger RNA, transfer RNA, it's going to carry out that information. Correct. Do you understand transcription and translation? If you don't, you got work to do. Yes? Let this diagram help you. Okay? On page 106 and 107 in your textbook. Kind of see it big and beautiful. Shows you all the little steps, all the little specifics. But what you're going to end up with is a polypeptide. Yes? And that is many amino acids in a very specific order based on what? That genetic code. And this diagram shows us that little knitting machine type thing that's going on there. So with the help of the ribosomes, we're going to get those things close enough together. Transfer RNA, messenger RNA, codons, anticodons are going to help bring together amino acids. And what kind of bond, here's a tricky chemistry question, is going to bring the two together? It's a peptide bond. Very good. What kind of synthesis? It's protein. Yeah, that's a good one. That's good. Think chemistry, though. What type of reaction? Say it out loud. Suck water out. Dehydration, synthesis reaction. Very good, very good, very good, very good. Oh my gosh, that was tiring. Okay, so this is the whole shebang all put together for you. Use the I know sometimes they can be a little overwhelming, but if you can sit down and very systematically look at the pictures and explain what's going on, that's helpful. What else is helpful? Yeah, studying is always helpful. Yeah, because all this stuff isn't just going to go bing. You know, it's not going to flow off your tongue. DNA unzips with the help of RNA polymerase. Messenger RNA strand is made. Then we're going to leave the nucleus and we're going to go to the ribosomes. We're going to dock at a ribosome. And transfer RNA is going to come along. Codon and anticodon are going to hook up. Through dehydration synthesis, a peptide bond is formed between two amino acids, and the whole unit moves over until the entire messenger RNA is read. See, that's... And then what's going to then we're going to alter it and do stuff to it. It's already in a set of canals, the endoplasmic reticulum, and then it's going to hand off to the Golgi apparatus for what? Packaging and shipping. Very good. Transport, yes? So as you can see in sort of what someone down here said, there's actually several different ribosomes that are working on one messenger RNA at any given time. And those pieces are all spliced together to create proteins. Then, in this diagram, we see what? Once we make that protein, that little chain of pearls, then we have to bring it someplace. So through these little transport vesicles that we find within the cell, we're going to bring it places. We might have to ship it out, and that's when Golgi's going to help me. We might have to maybe we have to just stick it on some of the membranes inside the cell. But it has to be brought to where it needs to do its work. Yes? And that little transport vesicle is going to help me do that. What am I doing? I'm going in the wrong direction. I guess I didn't drink enough coffee. All right. So information transfer from DNA to RNA to polypeptide is basically protein synthesis, is transcription and translation. So this little diagram shows us the specifics of messenger RNA, transfer RNA, codon, anticodon, and amino acids. There's a start message, and that particular message is always going to be start, and there's a stop. If you go back, I know you're going to kill me for doing this, but if you go back to this chart, you see the little green box over there? What does it say? Start. So AUG is start for every single protein. And it's a methionine amino acid. And then we have stop, and that can be several different ones depending on the protein. Those different codons are stop codons. Yes? No? Maybe? Sometimes? Start? Stop. How many different proteins in this? How many different amino acids in this protein? Well, there's 9, but one's a stop and one's a start. Yeah? What's 9 minus 2? Thank you. Yes? We good?
So they're always, for amino acid, there's always 3.
Correct. Nucleotides that will code for one amino acid.
Always 3.
And an anticodon is always 3. Yes? You understand transcription and translation? Yes, you do. You understand passive and active membrane transport. You understand all of the different terms we see in this table. You understand all of the different cell parts. Yes? These all look familiar to you because you have what? Studied, and you see them in the book. Yes? All right, let's try this, the possessed clicker. Any questions before we try the possessed clicker? Your exam is Tuesday of next week. Unless you have a really, 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 really good excuse, be here. Procrastinator people, no procrastinating for you. Be here. Ready or not, be here. Yes? Good. What are you going to do this weekend? You are going to study. Yes? And you are all, every single one of you in this class, going to get 100 on that exam. Everyone. Yeah, but if you all walk out of here with that attitude, hello? Yes? I'm going to get 100 on this test. Yes? I'm so getting 100 on this test. That's a better way to say it. Yes? Don't get this, oh, it's so hard. I have so much to do. Just do it. Stop whining. All right, the basic functional unit of living organisms. This is the clicker part. Let's hope it works. Five, 4, 3, 2, 1. Doesn't work. Son of a biscuit. Let's try that. I don't know what's wrong with this stupid thing. Oh well. Sorry guys. Doesn't work. What do you think the answer is? The answer is... Okay, what's going on? Well, maybe try this. Wait a minute. Nope, still doesn't work. Damn it. The answer is C. Who didn't get C? Remember, the smallest, what? What word in there? What's a key word in there? Living. If I say small, oh, there it is. That's awesome. Okay, that's good. All right, let's see if it goes away. Exciting. Okay. All right, the three main components of all cells, including the plasma membrane, the nucleus, and the... Oh, wait a minute. I got to push the what's this. There we go. Now here's the tricky to this one. All cells. All cells. Did we get everything? Everybody? Ooh. What's the answer, do you think? Very good. Main, now think main, like lots of main. Yes? Now how do I figure out the right answer? Okay. Oh, well, that's still going. This thing is possessed. I will figure it out. Well, at least you're all clicking in, so we hopefully will get some answers. Get rid of that. Why won't it go away? It won't go away. Oh, help us. Oh, wait. Phospholipids orient themselves in aqueous solutions such that what? You can't read. I know you can't. No, can't do anything to it. Control. I don't want to do that because I'm afraid. Ew. Let's end this clickerness. I don't know what I did. I just pressed. But I don't want to mess up my recording either, you know. I do, but see, nothing happens. When I hit the graph next to it, this thingy comes up. And for some reason, it stopped the clock last time. It's possessed. No. Oh, well. Okay. Oh, there we go. Well, at least we got out of there, but now we can't get the damn chart out of the way. Wait a minute. Let's try this. We'll kill it. Ah! We killed it. All right. Well, at least we have some clicker information. I'll try to figure out what the heck is going on. But, all right. A couple more of these, and then we'll go on to Chapter 4. A cell might increase its membrane fluidity or flexibility by increasing the amount of blank in the cell membrane. Now remember, this is the membrane itself, allowing things to go in and out. Yeah, we don't have clickers, so forget that. Trying to do the clicker. Yeah, it's unsaturated fatty acids. I wouldn't ask you that question. That's horrible. In areas of the body subject to a higher degree of mechanical stress, which of the following types of membrane junctions would you expect to be most prevalent? Stress, pull. What do you think? You think it's C? Those spot welds, that really tight stuff. Yeah. Yeah, well, when we talk about the desmosomes, yeah. Based on your knowledge of the cell membrane's chemistry, where would steroid hormones most likely interact with their target cell? Oh, you don't know that right now. Anyway, it's inside the nucleus. Don't worry about that. Bad question. Red blood cell placed, now you should know this. Red blood cell placed in a container of distilled water will what? Through what? Gain through osmosis, you think? You think, is that consensus? Very good. Which of the following is not an example of simple diffusion? What do you think? D? Think it's D? You're right. Everything else just happens through simple diffusion, movement of molecules banging into each other. And one more. When movement of sodium ions down their concentration gradient, what the heck does that mean? From high concentration to low concentration, drives the transport of other substances across the cell membrane, it is called and the key word here is drives, which implies that there's what? Energy being used. Okay? Questions. Give me questions. Questions? No questions on chapter 1, 2, or 3. Yes? Yep. First test and first test only. Multiple choice. All of it. First test and first test only. Why do you think I do that? To ease you into the test-taking process. Yes? Yes. Yes. I use the bubble sheets. Hey. Do everybody know what a bubble sheet is? Yeah, it's a little, you fill in the dots thing with your pencil. Do you ever take SATs, PSATs? Bubble sheet, yes? So what do you have to bring to class with you? Bring a pencil. I will have extras, so don't panic if you forget your pencil. But bring a pencil. What else? Huh? After that test, we will have combination tests, mostly multiple choice and essay questions. Ew. Imagine me, I have to correct them all. Like I say. Ew. Now, questions about material.
Oh, because one of the questions, like, not all cells have DNA.
No.
So, like,
All human cells do. Mostly all human cells do. Except for what? Red blood cells.
I was thinking about, like, sort of a big cell. Like, not all cells have the same organelles and stuff.
Correct. But they all have what? Cytoplasm. Question. No questions. Everybody knows all the organic molecules. Everybody knows the building blocks of carbohydrates. What are the building blocks of carbohydrates? What are the building blocks of carbohydrates? Not amino acid. No clay. No, not No! Carbohydrates are made of glucose, simple sugar. Glycerol and fatty acids. What are proteins made of? Amino acids. What are nucleic acids made of? Nucleotides, which consist of phosphate, sugar, nitrogenous base. Yes? Carbohydrates are not made of proteins. No. No. That seems to be the answer that comes out of everybody's mouth when I ask the proteins. It's like, no. Nope. So iron those out in your head. So how are you going to iron them out in your head? Yeah, you're going. But don't just read the book. Now you're going to iron it out in your head. So what's it going to take for you to remember that? Maybe you need to, do your little cheat notes. Maybe you need to write it down so you can see it. Maybe you may need to make your flashcard, yes? But you've got to transfer the information from here to here somehow. Yes? And just listening to me and then going, oh yeah, that doesn't do it. It's carbohydrates, lipids, proteins, nucleic acids. And there's 4 different what? What do we call them? What are all the strange things I just said? What are they on the whole called? Organic molecules. And why are they organic? Pray tell. They all contain carbon. Name me an inorganic molecule. Salt. H2O. Why are they inorganic? No carbon. What kind of bonds do they tend to have? Inorganic. What kind of bonds do they tend to have? Salts. Ionic bonds. Water is a covalently bonded inorganic molecule. What kind of covalent bond? Polar covalent. What makes it polar? Slightly negative, slightly positive end. It's an unequal sharing of electrons. What's an atom made of? Protons, neutrons, and electrons. And on the periodic table, the atomic number means what? Number of protons. The number of protons defines me as an element. Who has 1 proton? Hydrogen. Can hydrogen have different numbers of other subatomic particles? It's always still hydrogen if it has 1 what? What are all those other guys, those imposters called? Isotopes. Yes? Well, good. That's good. I'm excited. You excited? Questions? Any more questions? Question? Did everybody sign the sign-in sheet? Just in case? Yeah, make sure. Who's got it? Pass it up yonder, back in that region. All right. It's time to start the next chapter. What's the next chapter called? Tissues. It's the next what? Level of organization. Exactly. So we had chapter 2, chemicals. Yes?
We have a chapter 3 quiz that we need to do.
Yes. Oh, thank you for reminding me. I'm in the twilight zone. I apologize. And I don't know what the heck I did with your chapter 3 quiz, but it's up and it's due on Tuesday. Okay? It's open. It's ready to go. Say it again, hon. I know, I don't know what the heck I did. But it's weird. I thought. Did I emailed you back and I said it's, so I changed it last night. Something was weird last night. But when I emailed you, I put that time in. And why didn't it come in? I don't know. No. Do that because, you know, then I go in and check it. So it's there. Yep. And it's due Tuesday. I don't even know. Go check. I know. Well, do it before class, yes. Okay, so chapter 4. So chapter 4 talks about tissues. So now we're starting to get bigger. What you're going to see in this textbook is we're going to work through the levels of organization, and then once we hit the organs, that's going to be all your different chapters. Because the organs are going to be part of different systems. Everybody knows all the systems and their functions, right? Because that's going to be on the exam too. So the nervous tissue, muscle tissue, epithelial tissue, connective tissue are all different arrangements of cells that are going to help with all of the things on this diagram. So nervous tissue is part of what system? The nervous system. And in the nervous system, we're going to see different types of cells and different arrangements that are going to do what for us? Transmit messages, help to control different body systems to maintain what? Homeostasis. See, you guys know this. You know a lot. You know a lot more than you think you do. You'd be surprised. So, in this chapter, we're going to look at cellular organization and describe some of these different tissues. Part of anatomy and physiology is understanding the parts from their building blocks. And those building blocks are groups of cells. So it's extremely important for you to be able to recognize the cellular components of all the different organ systems you are going to study in an anatomy and physiology class. And when you look at these things under the microscope, and you've seen some examples already, you'll see some cells this week if you already haven't had lab. Those cells are beautiful colors, are they not? Are they like that in real life? No, they don't have beautiful colors. They're actually clear. Why is it we can see them under the microscope with all the beautiful colors? Yeah, we put them through a chemical process called the staining process. And that, based on chemical composition, is going to bring out all of the important parts of our cells. So we make a very thin slice of a group of cells, tissue, organ, whatever it is I want to be looking at under the microscope. And I stick it to a glass slide. Then I put the stain on it, let the cells absorb it, cover it up with a little glass cover slip, and that's what you are looking at. Somebody already did that for you. So, the first group of cells are very numerous in the body. The first group of cells that create epithelial tissues can be found all over the body. Epithelial tissues are extremely important. They cover things and they line things. So covering the whole outside of me is what? Epithelial tissue. Lining the inside of all of my blood vessels is epithelial tissue. Lining the inside of the 4 chambers of my heart, epithelial tissue. Now, depending on what we're talking about or where we're talking about, we can find different types of epithelial tissue. But when we look at epithelial tissue, there's very specific characteristics we have to keep in mind. Common characteristics, I should say. what we're going to see is a group of cells that are polar. What's that mean? No charges. Opposite things on either side. Okay? So, we have one side called the apical surface and the other side called the basal surface. So again, take these cells, put them all together, create a long line of cells. One side might be lining the inside of my blood vessels. The other side is going to be what? Stuck to something else so it can stay put. The basal surface is typically stuck to some sort of connective tissue. So when I look at an artery or in a vein, I'm going to find several different types of tissue. I'm going to find epithelial tissue lining the inside, stuck to a layer of connective tissue holding it on, stuck to something it's connecting to. Does anybody know what else is in a blood vessel? Muscle. Wrapped with some sort of membrane type material. So that's what an artery and a vein is. It's not just one type of cells. It's not just one type of tissue. Does that make sense? Okay. So we're going to, again, in this chapter, study all the different types of tissue and where I might find them. So another thing when I'm classifying epithelial tissue is I have to look at what it looks like. So I have an apical surface, I have a basal surface. I can have a single layer of these cells, or I can have a thick layer of these cells. The shape of the cells is also going to help classify them as well. So when I look at the cells, they can be simple, which is what? One single layer of cells, or they can be stratified, which is what? A whole bunch of layers of the cells. The shape of the cells can be different as well. They can be flat, sort of like floor tiles. We call that squamous. They can be kind of chubby and cube-shaped. What do we call that? Cuboidal. And then they can be taller cubes. What do we call that? Columnar. So you can have simple columnar epithelial cells, or you can have stratified columnar epithelial cells. Yes? You can have simple squamous epithelial cells, or you can have stratified squamous epithelial cells. Yes? Okay, good. So we have to understand size, shape, arrangement. And those are all going to help us with understanding and characterizing epithelial tissue. So, one side's going to be to lining something. With the case of my skin, one side's going to be where? On the surface, facing the outside. The other side is what? Stuck to some kind of what? Connective tissue, sticking it to something else. Yes? So, epithelial tissues support, extremely important for protection, for lining. We see exchange happening across them. When I talk about the lungs, for example, and I talk about exchange of respiratory gases, the epithelial cells are going to line those little balloons we're going to talk about next semester in the respiratory system called alveoli. And alveoli are the end of where your air goes. The alveoli are lined with lots and lots of little blood vessels called capillaries, which are also thin little epithelial layers. And air is going to come into your lungs, and oxygen is going to do what? Yeah, transfer, be transported across those couple cell layers into blood. Who's going to pick it up? Hemoglobin, and transport it, yes? When it picks up oxygen, it's going to drop off carbon dioxide. And what are you going to do with it? Exhale. Yes? So what kind of epithelial cells do you think I'm talking about? Stratified or simple? Really? Simple. If they were stratified, watch, wait. That just happened. All of that just happened. If it was stratified, it would take a whole heck of a long time to pass oxygen through a whole bunch of layers of cells, wouldn't it? See where I'm going? Good. So simple layers there. Who ate breakfast this morning? Oh, lots of hands. Yay! Look at all the bright faces. I do eat breakfast in your car. So bad. But yeah, I usually have my little smoothie that I make in the morning. It's yummy. It's got spinach in it. I'll give you the recipe. But this morning, out of spinach. Thank God. So I had to go to Dunkin' Donuts. And I get my little egg and cheese on my English muffin, being good. Sometimes I tend not to chew very well. A couple of chews and swallow. Your esophagus, what's that tube lead to? Your stomach is lined with epithelial cells. Guess what kind? Stratified. Because guess what happens every time you swallow? Your esophagus is about that big. When you swallow, what happens to it? It stretches. So my delicious egg and cheese on English is going to drag its way all the way down my esophagus into my stomach. So every time you swallow your egg and cheese, you also swallow what? The cells that it drags along with it as it goes down into your stomach. So if I had a simple layer of epithelial cells there, it would be very, very bad, yes? I would swallow once and I would be done, pretty much, right? You see the difference? So in places where you're going to get a lot of friction, like that outer layer of skin, which is many, 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 many layers of epithelial cells, versus that alveoli where quick stuff has to happen, you'll see simple versus stratified cells. Okay? And again, the shape has to do with the whole transport thing. Cell might have to grab things like make vesicles, endocytosis, exocytosis. So if I'm a little chubbier as a cell, I can do that a little bit better. If I need to have oxygen transport happen quick, if I'm flat, that can happen faster. So the shape of the cells we'll see in different places are based on their different functions. So the other thing about epithelial cells is because, my same swallow my breakfast example, every time I swallow something, I swallow the cells on the outer, what surface is facing the inside of tubes of an epithelial layer? The apical side. I'm sloughing those cells off. It would be nice if I replaced them, yes? So we're going to see that epithelial tissue regenerates itself very, very quickly. The other thing kind of strange about epithelial tissue is it's avascular. What the heck does that mean? No blood vessels. There's a blood supply, and it actually is going to help get food to the cells in the epithelial layers through the connective tissue underneath. And the simple layers get fed pretty well. But what happens to the stratified layers? They're getting food from underneath. What happens to that outer guy? You lose. So as they grow, we're going to see they grow from the bottom surface. We call it the basal surface. And the cells move their way up. the further they get away from their food source, what's going to start happening to them? They're going to start to die. So when we look at that outer layer of skin, it's actually made up of 5 layers of epithelial cells. And the outer layer, dead as a doornail. It's called the stratum corneum. So when I go like this, what happens? All those dead cells are falling off, and who's replacing them underneath? Yeah, the new ones are constantly being replaced. Same thing for hair. That's why it grows. Yes? Yeah. Mm-hmm.
It takes off several layers of cells, like a wall.
Yep.
When I peel that off my face, it actually feels wet and cold. Is that
Why? What's underneath your epithelial cells? Connective tissue. In the connective tissue, when we talk integumentary system, we're going to call epidermis, dermis. Dermis is connective tissue. Epidermis is what? That's all skin. But what's the epidermis? What kind of tissue? Epithelial tissue. All the nerves, blood vessels, they're down here in the dermis. So when I cut myself, sometimes you've probably done this. You scrape yourself and you don't bleed. What do you think you just cut off? Just the epithelial layer, though. The blood vessels are underneath. So when you peel off several layers, does your face start to hemorrhage? No.
No, it doesn't.
No. So you don't work your way down to what? You're not far enough to hit your dermis in your skin, but all of those nerves and blood vessels are much closer to the surface now. So that's where you get that cool sensation and you might get little tingling. It's because those nerve endings are firing off because they're closer to the surface. Yeah, maybe you shouldn't do that, is right. So, epithelial tissue regenerates itself very fast. Epithelial tissue is avascular. When I talk about epithelial tissue, I'm always going to talk about them as a layer. We're going to have cells, whether it be stratified or simple, that are connected to something, that basal membrane. The basal membrane is kind of like glue. It's created by the cells in connective tissue and the epithelial cells to stick it to what's underneath. And what's underneath? Connective tissue. So cells with basement membrane, that's a fancy name for that basilar layer, and what's underneath? Connective tissue. Yes? Good. So what's simple epithelia? Single layer of what type of cells? Squamous. Well, you can have cuboidal too, or you can have columnar. But simple is single. This particular tissue is simple squamous epithelial. So you have to have both in there, because we have to know how many layers and we have to know the shape of the cell. So it's kind of like floor tiles, basically. It's going to be squamous cells in a single layer with basement membrane and what's underneath? Connective tissue. And when we see simple squamous epithelial, we see some of these different functions. It's going to allow materials to pass fairly quickly from one side to the other. So diffusion and filtration, movement of stuff, is a place or is going to happen here. Where am I going to find it? In places like the kidneys glomeruli. What the hell is a glomeruli? That's second semester, almost at the end. What's a kidney do for a living? Filters what? It filters your blood. So the glomeruli is the beginning part of the little filtering units inside kidneys called the nephrons. Does there any material that has to pass from one side to another here? Yeah, from your blood to what? To the kidney through a little series of tubes that will eventually pass through your body as urine. We already talked about the lungs and those little sacs, and this is actually what we see here in the alveoli. So the cells we're talking about are actually right here. See them? They're kind of weird because take that floor tile that I just described, pretend it's my hand. Now take it this way and look at it that way. So it's very difficult sometimes when I'm looking at it this way to see all the cell parts. But you can see some of the nuclei in these cells sideways. Yes? Be easier if I had them this way. But they're not in this picture, they're this way. Okay, and what's these big spaces then? Those are the air sacs. And that's where gas exchange takes place between the alveoli and blood vessels. And unfortunately, there's not a blood vessel in here, which is too bad. The next type of cells, the next type of epithelial cells we're going to talk about is simple, which means what? Single layer, cuboidal, cube-shaped with basement membrane and connective tissue. Very good. These are lining tubes. Remember I said we're either going to line tubes or cover something? Well, these particular ones in this histology slide are lining tubes. See the tubes? See the cells? There's one. There's another one. There's another one. Whoa, that looks different. Why? Remember, I'm taking a tube lined with cells, and what am I doing with it? So you can see this. What am I doing? I'm going, taking a slice, very thin slice, putting it on a slide. Now, if the cell looks like this, and the nucleus is here, and I take a slice right about there, am I going to see the nucleus? No. So see? Some of them made it, some of them didn't into this particular tissue sample. That's how thin the tissue samples are. So those are simple cuboidal epithelial cells. Places where the cells have a little bit more work to do involved in making things and pushing things out. So we're going to see them. This is one of the groups of cells in kidney tubules, little parts of those little filtering units I was talking about. What we're going to talk about in this chapter 2 is glandular material. And what do glands secrete? Some of them can produce hormones, some of them can produce sweat, some of them can produce tears, some of them can produce oils. So those glandular type groups of cells tend to be simple cuboidal. Simple what is next? Simple columnar epithelial. So what do we have? Single layer of column-shaped cells with basement membrane and connective tissue. Very good. So bigger, fatter cells. In the picture, we see them right here. There's a nice big fat nucleus. In this particular picture, the cells have little extensions on them. See that up there? What's it called? Microvilli. It looks like little hairs almost. Didn't we talk about villi before? What's it for? It might wave and move things along the surface. In the case of this microvilli, it actually takes the surface area of that cell and makes it bigger. So if I was to take this cell and just have this be the top of the cell, versus this be the top of the cell? Which one has more surface area? So if I was able to pull my fingers out and stretch them out, they'd be about that long versus just this long. So in the intestines, where these guys are, it increases surface area. What happens in the small intestine, do you know? Digestion. We're going to digest, break foods down, and eventually absorb them into the circulatory system. So the more surface area I can have, the better. Gives me more of a chance to get more stuff into the circulatory system. The other thing I have to be careful of in those tubes is making sure things move along freely. Sometimes if it gets too dry or the region gets too dry, that's not going to be very good for me, is it? It'll pull against those cells and it'll kill them. So we also see these in tubes like this. This is called a goblet cell. Looks like a big crackly balloon almost. And you know what this makes? Mucus. So mucus is kind of slick and slippery, and it's going to allow things to pass through the tubes without damaging the cells. We talked about mucus when we talked about our tracheas. Remember our disgusting smoking talk? And you know what kind of cells line your trachea? Pseudostratified ciliated columnar epithelial cells. Say that 10 times fast. We're going to talk about those guys in a minute. So what the heck does that mean? She didn't tell us about that. She said simple or stratified. Where the heck is pseudo? What the heck does pseudo mean? False. These cells are actually a simple layer, but when I look at them under the microscope, it appears that there are more than one layer, right? But they're not. Because the cells are kind of weird shaped, the way they line up. So the apical end and the, what's this end? The basal end that connects to the basement membrane, it's actually one cell, and I can actually follow it down if I look really close. It skinnies out and sticks to the bottom. This one here sticks to the bottom. Yep, so they're long columns, but they're not stratified. They look like it, but they're not. So what do we call them? Pseudostratified. They're ciliated because they have what? Cilia on them. So pseudostratified ciliated columnar epithelial cells, where do they live? In your trachea. What's the cilia for? To move along. One of the things you don't see in this are goblet cells. Those are there too. Similar to what we saw in the small intestine. Those are the mucus-producing cells. But they'll push mucus out onto the apical surface of these epithelial cells. Everybody understands what I just said, right? What's the apical surface? The one on the top, the one that's lining the tube or facing what's in the tube it happens to be surrounding. Who's this? Stratified squamous epithelium. What is it? Many layers of those flat tile cells with basement membrane and connective tissue. Very good. Now these are where? The esophagus. I'm going to see a couple of different types of these stratified layers. When we look at skin, it's going to look a little bit different, but very similar. The cells of this layer, the basal layer, is where the cells are born. And they will, as they grow, push their neighbor over and up. So as the cells grow from this layer, they push the ones that have already been made up in this direction towards the apical surface. They start to look different as they get around here, right? What happens to them as they get further away from their food source? They're going to start to die off. So they actually start to flatten out a little bit more. And what else? Do you see? Yeah, the nucleus starts to degrade. They start to die. So you can't see the nice chubby nuclei up in here. They start to get squished. So those are in places where there's going to be a lot of friction going on. There's keratinized and non-keratinized. When we talk about the integumentary system, we're going to be introduced to keratin. And what is that?
Protein.
Yes, it is a protein. And what's it made of? Amino acids. Very good. And it's a waterproofer. So when we talk about stratified squamous epithelium that lives out here, it also contains keratin, and that's going to help waterproof you. So non-keratinized places like on the inside. Then we have something called transitional epithelium. What happens with these guys is they start out one shape and they end out another shape. Is this a stratified or simple layer? Stratified. Basement membrane right here. Connective tissue down here. Cells here. So they're born here. They're going to do what? As they are born, they're going to push their neighbor up. And as we get to the top layers, what happens to the size of them? Yeah, they start getting stretched out and big. In places that stretch out, we're going to find transitional epithelium. Places like your bladder. What's your bladder do for a living? It's kind of a holding tank. Yes? If you didn't have a bladder, what would you be doing? You would be peeing everywhere all the time. Yes? Not good. So the bladder is going to collect materials that are being filtered from the kidneys. So when the bladder is empty versus when the bladder is full, and some of you probably have a full bladder right now, and they're saying, hurry up and finish so I can go empty it, it stretches. So we see transitional epithelial cells. We'll pick up with that on Thursday of next week. Do not forget you have a test on Tuesday. Don't forget to sign the sign.